Okay, could somebody confirm you can hear me?
Yeah, we can hear you.
Great. Before we begin, are there any questions? All right, I'm not hearing anything, so let me state that I've got some grading done. I got extra credit study 2 and extra credit study 4 graded. Hope to have extra credit study 3 graded, but didn't quite make it. So I'll get that one done maybe tonight. And then I did a worksheet for the lecture. You see what that is, worksheet for lecture 3. That's been graded. And worksheet for lab 4 and worksheet for lab 5 have been graded the first time. The only lab that's been graded twice, I think, is lab 1. Lab 00. Why haven't I graded that? Let me check that. Yeah, for some reason I don't have lab 00 graded for a second time yet. All right, any questions about the grading? Let me close down the grade book and then I'll share my screen. All right, you should be seeing my screen now. So today is October 22nd, and we should be finishing chapter 4. We're a little behind schedule, so I don't think we'll quite finish chapter 4, but we'll get close. We won't talk about the characteristics of eukaryotic cells with the exception of the endosymbiotic theory. And then in the lab today, we'll discuss and start the unknown project, which I'm not quite ready to send that out, but hopefully I'll get that finished tonight. Any questions about what we're doing? If not, let's jump into the lecture. So I talked about the passive processes, talking about both simple and facilitated diffusion, or the diffusion of solutes down a concentration gradient. Osmosis is the net movement of water down its concentration gradient, meaning water moves from areas of high concentration to areas of low concentration. But this is not either diffusion or facilitated diffusion because water is a solvent and we call it osmosis. Osmosis also involves the flow of water across a membrane. If you don't have a membrane there, it is simply the diffusion of water. But if it's across the membrane and moves from high concentration to low concentration, we call it osmosis. Oh, too far. Sorry. All right, when we're talking about osmosis, a bacterial cell can experience three different types of osmotic solutions. The cell can be in an isotonic solution, it can be in a hypotonic solution, and it can be in a hypertonic solution. Each of these terms is a relative term, so it's correct to say the solution outside the cell was hypertonic relative to the solution inside the cell. Any question about any of that? So when we say a cell is an isotonic solution, it means the concentration outside the cell is equal to the concentration inside the cell. And we discuss tonicity as the net movement of water, but the tonicity is measured by the solute concentration inside the membrane and outside the membrane. So a isotonic solution is one where the solute concentration outside the cell is equal to the solute concentration inside the cell. And that means there's no net movement of water into or out of the cell. So you could say the net movement of water is nil. In reality, it means that for each molecule of water that moves in the cell, there's one that moves out of the cell. The cell is happy in an isotonic solution because it doesn't lose or gain water, and the cell can remain alive. In a hypotonic solution, the solute concentration outside the cell is less or lower than the concentration inside the cell. Let me blow this up. So there's a net movement of water into the cell. You note when there's less solute concentration outside the cell, that means there's more water concentration outside the cell. So the water moves to areas of low concentration and water will move into the cell. That will swell the cell so that the cell membrane will expand as more water moves into the cell. If the cell have a cell wall, like a red blood cell, the water will move into the cell until the cell becomes puffy like a, I don't know, stuffed donut or a balloon. And then you continue moving water into the cell membrane will burst, like blowing too much air into a balloon. So hypotonic solutions are dangerous for the cell if the cell lacks a cell wall or the cell wall is damaged. It can be damaged either by penicillin, like penicillin G, or by an enzyme lysozyme. If the cell wall is damaged, then the cell membrane will expand out of the holes in the cell wall, causing the cell to burst, and that we call osmotic lysis. Most bacteria live in a hypotonic solution, and that's why penicillin G can damage the cell. Any question about any of that? A hypertonic solution was where the solute concentration is higher outside the cell and lower inside the cell. So there's more water outside the cell than, excuse me, there's more water inside the cell than outside the cell. So the net movement of water is out of the cell. This will cause shrinkage of the cytoplasm. It'll pull away from the cell wall, resulting in plasmolysis. This generally leads to cell death, depending on how sensitive the cell is. Like E. coli is very sensitive to plasmolysis. And after being in a hypertonic solution for, I don't know, just a few hours or a few minutes, uh, the E. coli will die. This is also why when you have molasses and you store it at room temperature, it can survive for years at room temperature because there's less water outside the cell and a bacteria falls into the molasses. The water leaves the bacteria, causing the cell to undergo plasmolysis and eventually die. Any question about any of that? I can tell you my mother had a bottle of molasses that we figured was at least 30 years old, and it never went bad. It hadn't been used since 20 years old. I once made my father molasses candy out of it, and it was perfectly good at 20 years old. My father ate the candy and enjoyed it. All right. Active transport is good when the movement of material is higher outside the cell than inside the cell, so that a passive process can bring the material into the cell. But what about when a cell is in an environment with a low nutrient concentration? For example, a cell in the ocean or a cell in the desert. How can they get the nutrients they need? It cannot get it from a passive transport because the cell will have higher concentration of nutrients inside the cell than is outside the cell. In these cases, a cell has to rely on active transport, the movement of material against a concentration gradient. This requires the cell to spend energy because the cell needs to perform work to move a molecule at low concentration outside the cell, moving it into the cell where it is at higher concentration. This is moving away from the way diffusion normally works. Cells use active transport to pump these molecules against its concentration gradient and to pump the molecule into the cell. This process, active transport, requires a transport protein in the cell membrane. Generally, a transport protein is specific for moving one and only one molecule into the cell. For example, there is an active transport protein to move glucose into the cell, and it only moves glucose. It doesn't move another sugar like fructose or beta-galactose. There is another active transport protein for moving um, beta-galactose into the cell. And then to move that molecule into the cell requires the cell to expend energy. What the cell does is it degrades ATP using the energy released from the third phosphate group coming off of ATP. So movement of one molecule into the cell by active transport uses up one ATP in the cell. Any question about any of that? Let me shut that down. All right. There is a special active transport that's found only in prokaryotes. We call it group translocation. It is an active transport in that the molecule moves into the cell against its concentration gradient. It does require a transport protein. It does require a high energy phosphate compound, but generally not ATP. An example is PEP, which probably doesn't mean a whole lot to you, but this is a phosphate group which comes off of the, I don't know, the PE. And then that phosphate group is transferred to the molecule that is moved into the cell. So you move glucose with group translocation, and then the transport molecule is chemically altered. The phosphate from PEP leaves PEP and then binds to the sugar in a covalent chemical bond. It's thought that this alteration of glucose prevents the escape from the cell and allows the cell to accumulate substances more easily. Let me explain that in the show here. Paint. In a eukaryote, we only have active transport. So if we have 100 glucose molecules outside the cell, and zero inside the cell, which wouldn't really be a good way to put that. So let's say 100 outside the cell. No, let's say 100 inside the cell and 50 outside the cell. It has to move against its concentration gradient, so that's why there's fewer outside the cell. Let me make that a little bigger. UK standing for eukaryote. All right, so the cell moves all 100 glucose molecules. I'm just going to go G into the cell by active transport. And the last glucose is moving from one outside the cell. Let's make another line here. One outside, and it's moving in where there are already 149 glucose inside the cell. So that is up. Movement is against a very steep concentration gradient. You guys all see that? You have one outside the cell and you're moving it inside the cell. And so that's movement against a very steep concentration gradient. Whereas in prokaryotes, let me move this over. I can't. So I'll just start it down here. If we have 100 glucose molecules inside and 50 outside, I can't select that. So let's see if that'll carry it. Nope. Glucose. All right, if we move 25 outside, and then move 25 into the cell, the last one to move into the cell is moving one outside. And well, let's go with group translocation. It's only going to be 24 inside the cell. So one moving into 24 is much less of a gradient than 1 over 149, which the eukaryotes have. Do you guys see that? Anyone not see that? And so it's thought that prokaryotes can have an easier time moving substances inside the cell because they can move about 50% of them by active transport and the other 50% by group translocation. And then it's much easier to move the molecule into the cell. Now, remember this glucose, which is moving into the cell here. There's 25 glucose 6 phosphates inside the cell. And I'd have to do that a little differently. 100 glucose inside, we have to make some of them glucose 6 phosphate. But you're kind of getting the point, and that is when you change it to glucose 6 phosphate from glucose, you then reduce the steep number by half. And I've got it 1 out of 24, but in reality, it would be like 50 glucose inside the cell and 50 glucose 6 phosphate. And then we'd say 50 glucose outside. And so instead of uh, 1 glucose outside, instead of 24 glucose phosphate inside, it's actually now 74. But still, 1 out of 74 is much less of a gradient than 1 out of 149. Does everybody see that? There we go. And the reason is because we're talking about glucose here, and then we'll have 1 out of 74 glucose 6 phosphate. So instead of having them all glucose being moved into the cell, half are being moved in as glucose, and then half are moved in as glucose 6 phosphate. Any question about any of that? It allows the cell to accumulate substances more easily than if the cell only had active transport to move stuff into the cell. And eukaryotes only have active transport, but prokaryotes have both active transport and group translocation. All right, let's turn to the cytoplasm. Cytoplasm is the stuff inside the cell membrane. It's mostly water, about 80%. It does have dissolved solutes in it. And then it does have stuff in it as well, like the DNA is dissolved in it, at least most of the time. The nucleosoid will be wherever the DNA is, the nucleosoid region. The ribosomes will be in the cytoplasm, but they do not dissolve in the cytoplasm. And then there's inclusions that are found in the cytoplasm. And the inclusion is stuff that has sedimented out of the cytoplasm. So there we can see an inclusion body there. There's one there. The ribosomes would be these little dots there. You can see a little bit of dots there. The nuclear area, called the nucleoid region in a prokaryote, is wherever the single chromosome of DNA is located. It is floating around in the cytoplasm, and most of the time it is dissolved inside the cytoplasm. The exception would be when the DNA or the chromosome of a eukaryote is undergoing mitosis or meiosis, and condenses. That may happen in prokaryotes. I don't know about prokaryote DNA replication, but anyways, at least in eukaryotes, the chromosome can condense and then it is no longer in solution. And then you can see, like the X chromosome, for example, that's because it's no longer in solution. Remember the nucleoid area, the nucleoid region of a prokaryote has no boundary with the cytoplasm. There is no barrier. There is no cell membrane in a, not cell membrane, nuclear membrane in a prokaryote cell. And it's wherever the DNA is floating in the cytoplasm. That's where we call the nucleoid region. It's not terribly important to know that it's different from the cytoplasm because most of the time it's wherever it's floating in the cytoplasm. There is no boundary. But when you stain it, the DNA tends to take on a different color than the cytoplasm. The ribosomes are the site where proteins are made. And this is such a big structure, protein and ribosomal RNA, that it cannot go into solution. It's too big to go into solution. And that's why we see them as dots in the cytoplasm because they're not dissolved in the cytoplasm. You should know that ribosomes are made up of subunits. You don't need to know the subunit names, but the ribosomes are made up of proteins, ribosomal proteins, and ribosomal rRNA or rRNA. Any question about that? Like I said, you don't need to know the makeup of the subunit. You should know the prokaryote subunit, the prokaryote ribosome is a little smaller than the eukaryote ribosome. A prokaryote ribosome is 70S in size. The eukaryote ribosome is 80S in size. The S is the way biochemists and microbiologists measure ribosomes, and they will measure them by putting them on an ultracentrifuge gradient and then spinning them in the ultracentrifuge. And the prokaryote ribosome will come to the 70S area, and the eukaryotes will move to the 80S area on the ultracentrifuge gradient. Any question about any of that? All you really need to know is prokaryote ribosomes are a little smaller and their size is 70S. And eukaryotes are a little larger and their size is 80S. Let's move to an inclusion. Inclusions are reserved deposits within the cytoplasm. This is something that the cell wants, and then the cell stores it. Initially, when the cell is accumulating the substances will be dissolved in the cytoplasm. But as the cell accumulates more and more of the substance, eventually enough of the substance will accumulate that it'll precipitate out of solution in the cytoplasm. When that happens, we call it an inclusion. Any question about any of that? There are different inclusions. You don't need to know the names of them or what they are. You just need to know that there are different inclusions. And it's just something the cell is accumulating. Like there's magnetosomes, that's an inclusion. These are sulfur granules. This is a cell that can metabolize sulfur, so it will, of course, accumulate sulfur in the cell. And the sulfur becomes so high in concentration that it begins to precipitate out of the cytoplasm, forming the sulfur granules. And then here's a metachromatic granule. So there, let me blow that up a little. There, marked in yellow, you can see the cell, and then it has three inclusions in it. Any question about that? All right. Another thing we need to talk about. Oops, that didn't work. Well, let's leave it there. Is an endospore. An endospore is a structure that some bacteria make to survive. So when the environment becomes unfavorable to the growth of the parent cell will begin to form an endospore. An endospore is a specialized resting cell produced mainly by bacteria, two families, Bacillus and Clostridium. There are, I think, a total of four families that can make endospores, but the other two families, I think they only have one member in it, one species in the family. So there's only one member in the whole family. But Bacillus and Clostridium have a number of families, a number of species in each family. The endospore is highly resistant to desiccation, heat, different chemicals, radiation, and generally the endospore can survive conditions that the vegetative cell cannot. The vegetative cell is the metabolically active growing form of the cell, which to begin with is the parent cell, which will then begin to form the endospore. So there we see the endospore, and there is the parent cell right here, making the endospore. When the parent cell dies, the endospore will be released and then can continue living. So the endospore is not a reproductive structure. You have one parent cell, it will die, and before it dies, it'll give rise to the endospore. So one cell makes one cell. It's not reproduction, it is survival. Sporulation or sporogenesis is the formation of the endospore, which the parent cell makes, which actually all of these that I can, well, not those, but most of these, like that cell there, is making an endospore by sporulation. And then there's germination, where the endospore will germinate and give rise to a vegetative cell. The vegetative state of the cell being one where the cell is metabolically active, growing and reproducing. The endospore is not growing, not reproducing, and to tell you the truth, it's essentially not metabolically active. Its metabolism is very, very slight. And that's why it can survive for so long in a harsh environment. Endospore formation by sporulation happens by the cell first dividing, and the DNA of one chromosome after division will move to one side of the cell. The second DNA after DNA replication will move to the other side of the cell, and then the cell splits into two, but the cell doesn't split evenly. There's a larger cell, which will be the continuation of the parent cell, shown here. If I can get my mouse to work, there it is. And then a smaller cell, which will form the endospore. The plasma membrane starts to surround the DNA in the cytoplasm, creating a independent little cell. And this will be an independent cell here. There'll be a continuation of the parent cell. The endospore then may move, depending on the species, move inside the cell. Some endospores happen at the very tip, so in which case it does not move. Some, it actually moves to the center, others off the center. This one is showing you off the center. And then the endospore forms a second cell membrane so that the endospore has two cell membranes. And then the peptidoglycan layer forms between the two membranes. And then a spore coat will form outside of the endospore, coating the endospore, and then making the endospore very resistant to almost everything, desiccation in particular. And then the parent cell will die, and that will release the endospore. Any question about any of that? Let's watch a little video. It's not real good, but it does show you making an endospore. There we go. After the commercial. There's no volume here, but so the endospore forming. Come back here. Blow this one up. So in this cell and all of this species, the endospore is actually forming in the middle of the parent cell. Sometimes they form off center, meaning towards the edge, but not right at the edge. And sometimes they do form right at the edge, depending on the species. All right, any question about endospores? If not, this cell is showing you the cell structures of eukaryotes. You'll note that there are differences. Yeah, let's see if I can shrink that. Nope. How do I shrink it? I don't know. I'm just going to blow this up. There are some different structures between plant cells and animal cells. Like plants have a central vacuole. In the plant cell, that is the largest vesicle. In an animal cell, they lack a central vacuole. The largest vesicle is the nucleus. Obviously, plant cells do have a nucleus. Animal cells do have a vacuole, but it is not a very large central vacuole. And that would be something like the perioxysome. There's only one vacuole, and that's the central vacuole shown here. But right there, that could be a vacuole, or that could be a vacuole. And a vacuole is just a vesicle that has stopped moving, and then it's storing the material for later use. So a vacuole is a vesicle that has changed its function and is now just storing the material. A vesicle is the same object, I guess, as a vacuole, except the vesicle is moving from one place in the cell to another. So a vesicle is transporting the material from one region to another, and a vacuole is just storing the material. And animal cells do have vacuoles, and and so do plant cells, and both also have vesicles. But only a plant cell has a central vacuole, and only an animal cell has a centrosome. The centrosome is not a membrane-bound organelle. It is a non-membrane-bound organelle. And it has the centriole in it, which is important for making the mitotic spindle in animal cells. Surprisingly, plant cells do have a mitotic spindle, but they do not have a centrosome. Plant cells have chloroplasts, animal cells do not. Let me see if I remember if there's anything that animal cells have that plant cells do not have. Off the top of my head, I don't remember anything that animal cells have that plants do not. But plants definitely have things that animal cells do not. And here's showing you an actual electron microscopic image of a plant cell here and an animal cell here. Some differences between a plant cell and an animal cell. Oh, sorry, it's not animal cell and plant cell. That's a eukaryote and a prokaryote. Should shrink that down so I can see that better. Yep. One difference I want to point out, I've already mentioned it, so let me point it out again. If the animal cell can move or swim in solution, it may do so from a flagella or a flagellum, or it can move from cilia, or it can move from pseudopods. For prokaryotes, the only way they can move is if the cell has a flagella, either a flagellum or several flagella. Any question about any of that? Let me briefly talk about the cytoskeleton. It is the cell structural framework, sort of the backbone of the cell. In eukaryotes, the cytoskeleton is very elaborate, like right here. The cell would be right here. And then there's the cytoskeleton. You can see, you can't even see the cell here. Here you can see the cell. And the cytoskeleton is either shown in red, green, or blue. The point being that eukaryotes have a very elaborate cytoskeleton, which essentially runs everywhere in the cytoplasm. Although remember, this is a protein filament, and in between the different proteins, you got space where the cytoplasm can be. Prokaryotes, the textbook actually has this wrong and has never corrected it. Prokaryotes do have a cytoskeleton. It's much more primitive than a eukaryote cytoskeleton, but there is a backbone in the cytoplasm made out of protein in the prokaryotes. And like I said, it's much more primitive, not as extensive. It doesn't run almost everywhere on this slide, almost everywhere in this slide. And in prokaryotes, it's much less extensive. It does help give the cytoplasm or the cell membrane some shape. But remember, in prokaryotes, the main determinant for the shape is the shape of the cell wall. Let me briefly mention the endomembrane system. In eukaryote cells, they have an endomembrane system, which is a transport system for moving things around in the cell. It involves three or four parts. First, there is the endoplasmic reticulum, which makes a protein on the rough ER, and then it can put those proteins in a vesicle, which will then bud out of the rough ER. And in the smooth ER, you don't make proteins, you make lipids, and the lipids can be budded up in a vesicle, which will bud out of the smooth ER. And then these vesicles transport the material from one part of the cell to another. Initially, they go to the Golgi apparatus, and then the vesicle fuses with the Golgi apparatus, delivering the contents to the Golgi apparatus. The Golgi apparatus can then modify the contents, like removing an amino acid from a protein, or what's another thing? Phosphorylating or glycosylating the protein. And then the protein will bud out of the Golgi apparatus in another vesicle. And then the vesicle will transport the protein to where the protein is supposed to be. Like if it's making a nuclear protein, the vesicle will fuse with the nuclear membrane of a eukaryote cell and then deliver the proteins to the nucleus. However, if the vesicle moves to a location and then stops moving and just stores the contents of the vesicle, we no longer call that a vesicle, we call that a vacuole. So a vacuole is storing the contents within the vacuole. And those can be just stored for later use. Any question about any of that? Anyways, that gives the endomembrane system, which includes the ER, the vesicle, the Golgi apparatus, a vesicle.
With so much division in our politics, Joe Kent is adding.
And then the last location where the protein or lipid ends up, which if it's storage, it'll just become a vacuole. But if it's going to be used, the vesicle will move to where the protein is needed. Let's start this.
Science. In this video, I'd like to talk about the endomembrane system. The endomembrane system consists of organelles that create, modify, and transport proteins and lipids throughout the cell. It consists of the nuclear membrane, rough and smooth ER, the Golgi apparatus, and different types of vesicles. First, ribosomes, which are protein factories of the cell, can be found on the rough ER. These ribosomes create proteins and then transfer these proteins into the ER, where they undergo modifications, such as folding. These modified proteins may be absorbed into the membrane of the ER or packaged into a vesicle that may travel to the Golgi apparatus. The smooth ER may transport lipids to the Golgi apparatus, also using vesicles. A vesicle is a relatively small membrane-bound enclosed sac that stores or transports substances. At the Golgi apparatus, the proteins are sorted, packaged, and tagged so that they can travel to the right place inside or out of the cell. The receiving side of the Golgi apparatus is called the cis face. The opposite side is called the trans face. The transport vesicles that formed at the ER travel to the cis face, fuse with it, and empty their contents into the Golgi apparatus. As the proteins and lipids travel through the Golgi apparatus, they undergo further modifications that allow them to be sorted. The most frequent modification is the addition of short chains of sugar molecules. These newly modified proteins and lipids are then tagged. These tags help the proteins arrive at their proper destinations. Finally, the modified and tagged proteins are packaged into vesicles that leave the transface of the Golgi apparatus. Some of these vesicles deposit their contents into other parts of the cell where they may be used, while others release their contents outside of the cell by diffusing into the cell membrane. So there we go, the endomembrane system. Thanks for watching, and Moo Math uploads a new math and science video every day.
All right, any question about any of that? If not, let's move to the last part. And that's the endosymbiotic theory. The endosymbiotic theory is this theory to explain how eukaryote cells got a mitochondria and a chloroplast. So how did the mitochondria and the chloroplast evolve in eukaryotes? Where did they come from? How did they originate? Well, the theory is that we had a long time ago a big proto-eukaryote, meaning a cell before a eukaryote cell. That's why we call it proto-eukaryote. I'm just going to abbreviate that P for proto, and then E for eukaryote. And then we had a bacteria which was outside the cell. I'm not drawing that very well. Let's try and make it. Trying to make it rod. It's hard doing that. There we go. Well, the proto-eukaryote engulfs the bacteria, probably by pseudopods. Meaning it puts out pseudopods here and here. And these pseudopods just grew and then grew. Make that a little bigger. Until the pseudopods surrounded the cell. And let's draw a little thing around this. And then the cell was enclosed in a vesicle, which was actually at one time part of the cell membrane of the proto-eukaryote. And then this moved inside the cell. So let's redraw this. Actually, it'd be easier just to draw it here, expand the cell. I don't have to move it. Now, initially, the proto-eukaryote did not consume the bacteria. So it, did not digest the bacteria, did not kill the bacteria and digest it. It just left it for later use, meaning a reserve food supply inside the cytoplasm. And the proto-eukaryote could only engage in fermentation to get its glucose, to get ATP from glucose. Sorry. So it got glucose. I'm not writing here. It got glucose and used it to make 2 ATP, which I don't think we've talked about fermentation yet, have we? I don't think we have. But fermentation only results in 2 ATP from a glucose. And that's all the proto-eukaryote could get out of 1 molecule of glucose, 2 ATP. But this bacteria could perform aerobic respiration. Make that a little bigger. And that meant from 1 glucose, it could make 38 ATP. Well, the two were together, and glucose could move across the vesicle membrane as well as the cell membrane. So the glucose diffused into the bacteria. And then the proto-eukaryote lost 1 glucose and the bacteria gained it. And the bacteria took the glucose and made 38 ATP. And then the bacteria released some of that ATP into the proto-eukaryote. And the reason for doing this is probably just that life was simple back then, and it wasn't keeping track of the molecules. And we don't know how many ATP the bacteria released into the proto-eukaryote, but it was a lot more than 2. Okay? And was probably something less than 38 ATP because the bacteria probably kept some of the ATP for its own use. And it's hard to say what it is. I'm just going to take it something like half. What is that, 19? So it released 19. And we don't know what that number was, what it released. Okay? But 19 were given up, or some number much greater than 2, but probably less than 38 ATP were released. And the proto-eukaryote was initially confused, saying, I lost 1 glucose, but instead of gaining 2 ATP, I gained a bunch of ATP. Bunch of ATP. I don't know what I did there. So the proto-eukaryote decided to keep that bacteria around and then feed it glucose because the bacteria could make more ATP than the proto-eukaryote could. So the two started living together in harmony, where the proto-eukaryote gave the bacteria a home, fed it glucose, and then the bacteria digested the glucose and then released ATP for the proto-eukaryote. And then the two lived in symbiosis for a long time. Nowadays, that should be I. See if I can change that. Yep. nowadays we don't find the bacteria anywhere else except in the cell of the proto-eukaryote. And now we don't call this a proto-eukaryote because it is a cell that has an organelle in it that makes ATP. And this organelle is believed to be the mitochondria. Any question about any of that? Let me go back to my slideshow. There we go. So it's believed that the mitochondria at one time was a free living bacteria that became engulfed in a precursor to the eukaryote cell. And then the two started living in symbiosis. And now we don't find the bacteria free living. And then the proto-eukaryote has that bacteria inside of it as the mitochondria, and we call that a eukaryote cell. And the strange thing is that happened in plants and animals where they gain the mitochondria, but in plants, later on, after they gained the mitochondria, they also gained a photosynthetic bacteria, which later evolved into the chloroplast. There is strong evidence for this theory. First, when we look at some primitive protists, they can engulf a bacteria, like this paramecium gained a photosynthetic bacteria that it engulfed, and then the protist, which is a paramecium, gives a home and feeds the bacterium, and then goes to the light so the bacterium can get light, and the bacteria then makes glucose, and then the bacteria releases some of that glucose for the paramecium. So we do see this happening. In fact, today there is one family of amoeba that actually lack a mitochondria. And these amoeba go out and find a bacteria and then engulf the bacteria and then live in symbiosis with the bacteria, meaning they give the bacteria a home, give it glucose, and then the bacteria makes ATP for the amoeba. So we actually do find some protists which are doing the story that we suggested happened to the original eukaryote cell, where it engulfed a cell which lived in symbiosis for a long time with the proto-eukaryote and evolved into the mitochondria, and then the proto-eukaryote evolved into the eukaryote cell. So we actually do see cells engulfing a bacteria, and then the bacteria makes ATP for the eukaryote cell, the amoeba, and the amoeba gives it a home and feeds it glucose. So that's the first evidence. The second evidence is actually shown to you right here, and that is if an organelle were to be, not an organelle, if a bacteria were to be engulfed by a eukaryote or a proto-eukaryote, the organelle would have two membranes. Where do I have that drawn here? So there would be the cell membrane shown here. I don't have the cell wall shown, but there's the cell membrane of the bacteria. And then this is the vesicle membrane of the organelle. And what do we find in mitochondria and chloroplasts? We find that these membranes have two membranes, an inner membrane and an outer membrane. One being the original cell membrane, the other being the vesicle membrane that surrounded the engulfed bacteria. So that's some evidence. Let me blow this up. Some other evidence is when we look at the DNA of a eukaryote cell, the DNA is linear. When we look at the DNA of a prokaryote, meaning a bacteria, the DNA is generally one circular DNA molecule. When we look at the DNA of mitochondria and chloroplast, it is one circular DNA molecule. When we look at the presence of histones on the DNA, in eukaryotes, there's histones. In the prokaryotes and the bacteria, there is no histones on the DNA. On the mitochondria and the chloroplast DNA, there is no histones. When we look at the first amino acid made in protein synthesis, in a eukaryote, the first amino acid made is methionine, meaning AUG codes for methionine. In bacteria, it does not code for that amino acid. It's a similar amino acid, but a different amino acid that we call formylmethionine. When we look at the first amino acid made in protein synthesis in a mitochondrial chloroplast, it is formylmethionine. If the mitochondria and the chloroplast were bacteria that were engulfed by a proto-eukaryote, you would expect to find ribosomes in these organelles. And the mitochondria and the chloroplast have ribosomes in them. Furthermore, the ribosome of a eukaryote cell is 80S, the ribosome of a bacteria is 70S in size, and the ribosome of a mitochondria and the chloroplast are 70S. When we look at the way the cells grow, eukaryote cells grow by mitosis, prokaryote cells grow generally by binary fission, where one cell splits evenly into two. The mitochondria and the chloroplast, they grow by binary fission. And then the strongest piece of evidence that the mitochondria and the chloroplast are bacteria is that when we sequence the DNA of a eukaryote cell, the DNA falls within the domain eukarya. When we sequence the DNA of a bacteria, the DNA falls within the domain bacteria. And then when we sequence the DNA of the mitochondria and the chloroplast, the DNA falls within the domain of bacteria. Any question about any of that? All right, so that's the theory of how eukaryotes obtain their mitochondria and their chloroplasts. These organelles are different from other organelles, then they have a DNA molecule in them, they have ribosomes in them, and these organelles have all the properties of a bacteria cell that became engulfed within a proto-eukaryote. Any question about that? Any question at all about chapter 4? All right. If there's no questions, I think I'll end this lesson a little bit early, and I will see you in the lab at 6:30.